Hi there. Today I'd like to talk about what I think of as the most important gene causing early onset Alzheimer's disease. And that would be the gene presenilin-1, or more commonly called PSEN1, because that's the shortened abbreviated name of the gene. When there are around 20,000 different genes in the human genome that encode proteins, we need to have shortened names for them because some of them can be quite long. I have mentioned the presenilin gene previously in my third video when I was talking about the mutations that cause familial early onset Alzheimer's disease. And I've also previously mentioned the amyloid precursor protein gene. So I thought it might be interesting to start by looking at the relative importance of these genes in terms of research activity. Now, keeping in mind that in that third video, I mentioned how important the presenilin-1 gene is as a site of mutations for early onset familial Alzheimer's disease. As I state down here, over 60% of all of the mutations causing early onset familial Alzheimer's disease occur in the gene presenilin-1. However, if we do a PubMed search for either presenilin or for amyloid precursor protein, what we find is that we only get about 7,600 results for presenilin, but we have nearly 59,000 results for amyloid precursor protein. So there's a great deal more interest in the amyloid precursor protein in terms of research. If we look at the history of publications since the first, apparently back near 1993, that mentions the term presenilin, then we can see that interest appears to have peaked at around 2015, whereas interest in amyloid precursor protein has been rising fairly steadily, although it may have indeed actually peaked recently. We'll have to see. To give you a short history of the research on the presenilin-1 gene, I can tell you that initially it was known as, for a short time at least, it was called S182, before ultimately becoming known as presenilin-1. It was discovered by multiple research groups to be linked to early onset familial Alzheimer's disease in 1995, so about 29 years ago. And then 4 years later, a number of research groups showed that presenilin was involved with gamma secretase activity. Now, this is the protein cutting activity that cuts the amyloid precursor protein and is one of the protein cuts required to produce the amyloid beta peptide, which I spoke to you about in the earlier video on APP. So there was great excitement about this because it was seen that the presenilin gene was actually cleaving the protein from which amyloid beta was derived. And today we recognize presenilin-1 as the most common site for mutations causing early onset familial Alzheimer's disease, or EOFAD as I like to call it. So I'm going to show you a couple of figures from my recent hypothesis paper in order to illustrate some of the ideas about presenilin-1. Here's figure 1 from that paper. It looks a little bit complex at first, just at first take, but all it's really showing you is how the amyloid precursor protein is cut up into various fragments, including the amyloid beta fragment that deposits in the brains of people with Alzheimer's disease, and also the brains of people without Alzheimer's disease. But anyway, if we start here in the middle, we can see the amyloid precursor protein, or at least a representation of it, and it's sitting within a membrane, as in it crosses a membrane. We have what we refer to as the N-terminal end of the protein up here, and the C-terminal end down here. And this here is meant to be the lipid bilayer. That's a a fat membrane that makes up cells. Cells contain a number of these membranes. Anyway, we can see that in order to produce the amyloid beta peptide here, we need to make sure we cut the amyloid precursor protein in a specific way. Firstly, we need to cut it with something called beta secretase, which cuts at this particular position here. Now, we don't want to cut it with something called alpha secretase. This is an alternative method of cutting the amyloid precursor protein. We don't want to cut it with that, because if we cut it with that, we're going to end up producing something called P3, and that's not amyloid beta. But if we cut with beta secretase, we can then cut it within the lipid bilayer using something called gamma secretase. Gamma secretase is actually a protein complex. It's a group of different proteins together. It's called the gamma secretase complex, and part of the gamma secretase complex, one member protein of that, is presenilin. Presenilin is actually, as you can see from this diagram, it spans the membrane multiple times and sits very much within this membrane, this lipid bilayer. And interestingly, it cuts proteins within the lipid bilayer, and this helps to free these proteins from the lipid bilayer so they can go out of a cell, for example, or into the interior of a cell, the cytosol. Those two little lightning bolts there represent the cutting of this particular fragment. This particular fragment of the amyloid precursor protein is called beta CTF, in other words, the beta C-terminal fragment, because it's the one that's derived from cleavage by beta secretase here, and it includes the C-terminus. So this is beta C-terminal fragment. And if you cut the beta C-terminal fragment within the lipid bilayer, you release the amyloid beta peptide. So there we have it. Now, one more thing to point out about this diagram and presenilin within it is that if you look here as I've represented it, the presenilin protein is divided into two parts. And we actually would call these the N-terminal fragment and the C-terminal fragment of presenilin. Now, presenilin is first produced as just one complete protein, but it seems that when it's first produced, it's not really active in gamma secretase activity. And what needs to happen is it needs to be cut into these two fragments, the N and the C-terminal fragment, that remain associated with each other within the gamma secretase complex. But when the protein is separated into two parts like this, it then becomes active in gamma secretase. And actually the presenilin is the particular member of the complex that does the cutting. It does the catalysis here of the proteins, what we would call the protein substrates, the substrates of the enzymatic activity, which is the gamma secretase enzymatic activity produced by this complex. Okay, a lot of sciencey words here, but that's just explaining the details of what's going on. Now, if we look closely at the presenilin protein using another figure from the paper, which is actually taken from a really fantastic website, which I encourage everyone to visit, called Alz Forum. Alz Forum is mainly a website for researchers on Alzheimer's disease. It's all a bit technical there, but they have some wonderful collections, databases, and some great diagrams of the different genes and proteins involved in Alzheimer's disease. It's also a place where research type news and views articles are written, and people are allowed to post comments on those articles, usually comments on the science. Now, this diagram ultimately comes from Al's forum, and I used it with permission in the paper I published. And what you can see is it shows the presenilin protein with each little circle here joined together. That's meant to be the peptide chain, the amino acids, or more correctly we would call them in this case amino acid residues, because they're no longer free amino acids, they're all joined to each other by peptide bonds. But so the amino acid residues all strung together into a protein, and as you can see it has its N-terminal end, the N-terminus, and it has the C-terminal end up here, and it winds its way through the lipid bilayer a number of times. It loops out, through the bilayer, looping in, looping out, and so forth. In and out, around, and then eventually getting to the C-terminus here. Now, this is a really interesting diagram because what it shows you is a number of features. For example, if you have a look down here at the color code you can see on the different amino acid residues, and if those amino acid residues are changed by mutations in the gene, and those changes turn out to be disease causing for Alzheimer's disease, then they have this kind of very dark red color. And you can probably see that a number of these amino acid residues are colored dark red. In fact, there's a great many of them, over 100 of them. And you can also see something interesting from a protein researcher's point of view. And that is, often when you find mutations in a particular protein causing a disease, often you'll see them, for example, those mutations will be concentrated in one part of the protein, a particular domain, as we would say, that has a particular function, because proteins can be built up of different overall structural blocks, which we call domains, and those domains can have different separate functions. But what you can see with the mutations that cause Alzheimer's disease in the presenilin gene, presenilin 1 at least, and the same is the story for the presenilin 2 gene, its relative, is that they're quite spread out throughout the protein. And yes, they are concentrated within the transmembrane domain regions, as we would call them, but they're not only there, they also occur outside in some of these loops. There are some areas that don't appear to contain the mutations, and one of those is the N-terminal domain. One of those is the C-terminal domain, and interestingly, actually, if you look at a lot of the transmembrane domain just before the C-terminus there, you can see that a lot of those amino acid residues are not changed in Alzheimer's disease. And actually that suggests to me that those residues may be somehow important for the mutant protein otherwise to have an effect. It's something I'll explain later on when I'm talking in more detail about the different ideas about how these mutations cause the actual disease, because that's something that really is open to debate, I think. Anyway, and also there's this thing which we call the cytosolic loop domain, because it's a very large loop in what we call the cytosol, which is the area inside the plasma membrane, but often say maybe outside of organelles, which are the different functional structures within the cell. You can see that there's very little mutation in here that causes Alzheimer's disease. Something else you can see here, you'll see there's a couple of arrows pointing to amino acid residues marked D. Now, those are the important, those are actually very critical amino acid residues. They are what we sometimes call the catalytic aspartate residues, within the protein, and they are very important for the actual catalytic action of this protein to cut for example, amyloid precursor protein gene here. Those, you can think of those lightning bolts here coming out of those aspartate residues to cut the protein. Okay, just a couple of other things to mention. While the great majority of mutations in this particular protein cause Alzheimer's disease, this is an amazing protein in my opinion, because there are other mutations in this protein that cause a completely different disease. They cause an inherited skin condition called acne inversa. And there is a very specific difference between those mutations and the Alzheimer's disease causing mutations, but that's something I'll talk about in a later video. It's a very interesting topic. So I'll finish this video by just leaving us with a couple of really interesting questions, but there are so many about presenilin 1. Why do so many mutations causing early onset familial Alzheimer's disease occur in just the presenilin 1 gene? What's so special about this gene? What do these mutations do to the protein produced by the gene that ultimately then causes Alzheimer's disease? We'll look at some of these questions and more in later videos. Until then,